So what will we do with the NADPH and ATP from the light-dependent reaction? Well, let's look into the Calvin cycle. Now, well, carbon will be synthesized within the stroma. Now, there are 3 stages in the Calvin cycle. Fixation. In fixation, we are taking CO2 plus RUBP, and it's being connected with a ribosco to form a 6-carbon molecule. The 6-carbon molecule, though, is unstable and breaks apart into 2, 3 PGA molecules. Now, we have these 2 molecules, and one will poof go out, and one will move down to the next step of the process. And that one moving out goes to the cytoplasm. If I could had a green, I would draw green for cytoplasm. So the next step is reduction. So in reduction, we are taking some of those ATP and NADPH electron carriers from our light-dependent reaction and changing that 3 PGA into G3P. Now note, as I just said, that we're taking the products from our light-dependent reaction. So without the light-dependent reaction, the Calvin cycle will sooner or later stop because no electron carriers are being produced from the light-dependent reactions. So our next step with that G3P, we are going to get the regeneration step. Now on the regeneration step, our G3P, it is going to become RUBP. Now, I mentioned it, but I did not write it out, but RUBP is used during that fixation. So what happens, that regeneration, this comes back up. And as you see, it's a cycle. Now, just to bring this all home, at the end of the, say, light-dependent reaction, it produced and excreted 6 molecules of O2, and during the Calvin cycle, overall ended up producing sugar, our C6H12 of 6, which is our molecules needed in order to do the respiration, cellular respiration.